The Fibrobesity program at the University of Oulu looks at the interplay between obesity and tissue fibrosis. And we also try to look at the possibility of targeting tissue fibrosis in order to reverse this chronic disease.
So both obesity and fibrosis are known to affect fertility quite badly and in detrimental ways. So where it's rather well established that obesity is having effect in fertility in all the possible ways. There is very little known about the function of the fibrosis there, and especially what they do together and which one comes first, fibrosis or obesity, or both are affecting at the same time. My lab is focusing on three major questions in the field of this research program. So we're asking questions how uterine glands are forming, how they're affecting extracellular matrix. We are also addressing and developing new technologies, how to study extracellular matrix during early implantation stages, as well as developing new methods to address fertility and find biomarkers related to uterine receptivity. It's not known that in such process as pregnancy, actually collagen accumulation is critical for pregnancy establishment, and that would be called now normal or so-called also physiological fibrosis. So when establishing these terms, that's not the only part. So further things we noticed that actually methodologies are lacking out there, and to study, especially during early pregnancy, how accumulation of extracellular matrix, namely collagen, is taking place, and that's where our lab took a role to go on to establish new methodologies. Having diagnostics and also treatment options provided by molecular means will encourage solving these problems early on. Another important part that nowadays IVF can help to conceive, but also the same disease can be carried to next generation. We're looking at the same time for the ways how we can prevent the passing of the disease to another generation, and molecular methods can help in the future to address that as well.
In my lab, we are looking at the ovarian cancer and the link between ovarian cancer and omentum metastases. Now, ovarian cancer is the deadliest cancer because initial stages of ovarian cancer are asymptomatic, usually develops in women around 50 or later. We don't have biomarkers for screening, and when the patient comes to the clinic with ovarian cancer, it's usually in the stage 3 and 4 where metastatic development has already occurred. Also, ovarian cancer is one of the cancers that it's very characteristic for drug resistance. So it means that when we treat a tumor with drugs, a majority of cancer cells will respond to that treatment and will die. However, there is always a small population of cancer cells that are not responding to the drugs and they are not dying. And this small population of cancer cells will later start to proliferate, and also they can migrate to the other sites in the body and can initiate the development of metastases. And usually metastases are characterized by drug resistance. So one of the features, characteristics for ovarian cancer is that the metastases happen within the omentum. So omentum is the natural fat layer that we have enveloping the peritoneal cavity. And it has a lot of good functions, immune function, and it protects the inner organs. However, the ovarian cancer cells, they like to migrate from the primary site into the metastatic omentum site. And once they go to the omentum, they actually also develop fibrosis. So these cancer cells will start proliferate, and the tissue around the cancer cells will start to harden and develop this very stiff extracellular matrix. Why is this important for the treatment? It's because usually when the metastatic tumor is enveloped by this hard matrix, the drugs are not penetrating inside the tumors. So clinically, it makes it very hard to kill those tumors. In my lab, we are actually working directly with patient samples. So we get, for example, tumor biopsies, or we get small tumors from the surgery, and we get it into the lab, and we are looking at the biological properties of those cells. Normally, we culture them in different conditions, being in 2D or 3D. We look how the cells are proliferating, how they are migrating, and also we do high throughput drug screening. So it means that we have libraries of different drugs, and we apply those drugs on the cancer cells, and we looked at what are the most important drugs or the most effective drugs that are killing those cancer cells. We actually have been able to uncover new drug resistance mechanisms, for example, in ovarian cancer. And we have also been able to understand how the cancer cells like to develop metastases within the omentum, and also understand the fibrosis, understand the extracellular matrix role in supporting the metastatic development, and what drugs or drug combinations we can use to target this tumor microenvironment. So I think that we have to definitely be very optimistic for the future, and I think that the current research strategies that are employed in Finland and also in other countries are actually very competitive and very close to clinical settings.